Thank you very much. And what I want to do today is, this is going to be a very informal lecture. So if you have any questions as we go along, just raise your hand. And that's what Steve is here for. Steve will answer the questions. But seriously, I'm really delighted that we can be here and let you know a little bit about what the Bar Shop Institute does. I'm focusing tonight on an area that is, I think it's going to be critically important in the future, and that's Alzheimer's. And this is because Alzheimer's disease already has a major impact on our society. It's estimated that approximately, currently, annually, we spend more than $100 billion on this disease. It's the fourth leading cause of death after heart disease, cancer, and stroke. The real thing that's shocking is the amount of cost to a family, and it's estimated that it's over $200,000 of a family that's affected by Alzheimer's. And third, approximately up to 30% of the individuals in a nursing home can have Alzheimer's. I know this personally because my mother was diagnosed with Alzheimer's or dementia about 3 years ago. And she's now 96, but at that time she was able to live in her home and be independent because my brother lived very closely. But as the disease progressed, we basically had to put her in a nursing home because there was no other option. If it hadn't been for this, I think she still would be able to be in her home with some care. But this is one of the things that you can't get out of almost going to nursing home with this without having all-time care at your home. Now, the other scary thing about this is that this disease is going to almost quadruple in the next 40 to 50 years. And the reason for this is not that there's an epidemic, but it's because of the fact that there's more and more older people. And as you can see by these circles, is that the orange part is the number of people that are over 85 years of age. And you can see this is increasing dramatically. And this is what's going to drive this huge increase. So that you can see that we're talking about $100 billion annually now. In 40 to 50 years, we're talking about $400 to $500 billion if we don't do anything about this disease. Now, what causes Alzheimer's? Well, actually, I would say that we don't really know for sure. We know that one of the hallmarks of Alzheimer's disease is the presence of these plaques that are kind of in the that you see here. They're kind of orangish under this particular stain, and these tangles. These occur in various regions of the brain, particularly those parts of the brain that's involved in memory. And so it's generally believed that these plaques build up and they kill off the neurons, and this is the reason that you have the loss of memory. As I said, this is one of the most popular theories of how Alzheimer's occurs, but there are several people that question if these plaques and tangles are the culprit that causes this. But what we know is that as this disease progresses, there is the loss of memory until it gets so severe that the people even forget how to eat and swallow, oftentimes, if it goes to its extreme case. Okay, now, the risk factors in Alzheimer's disease. Steve will like this because I swiped this graph from him. So you can see things like physical inactivity you're talking about, it increases your risk about 30%. This gene ApoE is a variant that can, if you have that gene, it increases your risk 80%. But the key here that I want you to realize is the major risk factor for Alzheimer's is aging. And you can see here, this shows the relationship to the risk for age compared to these others. They completely disappeared. It's about 1,000 times greater than the others. So that if you look at the progression of this disease, in other words, the prevalence as a function of age in our population, you can see that before 65 years of age, Alzheimer's is very rare. Those cases that you see are genetic cases that you see. As you increase in age, you can see that not only it increases, but it increases dramatic. And it appears, I've asked my colleagues that do research in this area, that if you go even further, it looks like it keeps going up and up and up. So what does this mean? That if you're fortunate to make it to 90, 95 years of age, your risk of getting Alzheimer's is almost 50-50. And so that as you're going through there, it has a major, major impact on quality of health. So one of the things that we've been very interested in is what would happen if you slow down aging. Remember from this graph that age is the most important risk factor. If we were able to slow down aging, what would happen? Now let me ask you folks, do you think that we can slow down aging? Is it possible to slow down aging?
Give me a retirement at the same time.
Yeah, okay, right. You still want to retire. You don't want to push that back. So we don't know with human beings, but we certainly know with animals that we can slow down the aging process. And one of the manipulations that we can do this with is caloric restriction or dietary restriction. And this is a discovery that was largely made at the Health Science Center. It wasn't made at the Health Science Center, but in the last 30 to 40 years, we've been the pioneers in this area. And what you can see here, this is showing the lifespan of mice. We're talking about mice here. And this shows the lifespan, so the age. So the survival curve, as you can see in the black here, these are normal mice that are fed normal chow and this sort of thing. And they have an average life expectancy of around 25 to 30 months of age. And all of them are dead by about 38 months of age. Now, if we take the same genetic strain of mice, and roughly at about 4 to 5 months of age, we put them on a diet so that they have approximately 40% less food. They just don't get quite as fat and they don't grow quite as big. If we do this, we have a 25 to 30% increase in lifespan here. And you can see here, what's striking here is when all of these animals have died here, we still have about 50 to 60% of the other animals alive. In other words, if we were talking, if we could do this in human beings, we would be talking about, instead of an average life expectancy now of approximately 80 years of age, we would be talking about 100 to 120 years of age, average life expectancy. Now, one of the things people will often say, you're a nice audience because you're not like that, but some of my audience will say, well, we don't need to live longer. We've got too many old people around. But the key here is that because we've had this increase in maximum lifespan, we think that this process, the animals are living longer because they're aging more slowly. And the key here is that this manipulation improves the health span, or the health of the animals. The animals actually appear younger. And I'll let you folks judge for yourself, because I would guess that you're pretty good at dissecting the age of mice, right? Have any of you seen old and young? Well, I'll help you out here. Okay, so this is a video clip. These mice would be 5 months of age, which I like to say are kind of like college age humans. In other words, this is right when the peak of sexual activity and their peak of performance is, roughly. So they're like college age. Now, we're going to take the next thing, video clip, shows these are animals that are like about a 70 to 80-year-old person. And you can see that immediately the first thing is they're not moving around as fast. Now, these animals don't lose hair, but you can see their coat is more scruffy. And the other key is they're always looking for where they put their glasses. So there you've got a feeling. Now, the next clip I'm going to show you is the same aged animals, but these were the caloric restricted animals, the animals that live longer. And you can judge whether you think these animals look younger or not. So you can see immediately that they're more active. Their fur looks much younger. And so everything that you would look at, these animals are living longer and they're living healthier, at least as this view. The other important thing is that when we look at caloric restriction, it has a major effect on retarding or reducing most, if not all, age-related diseases. So in mice, we've shown that it retards or delays cancer, atherosclerosis, and a variety of things. And what I want to stress today is it even has an effect on Alzheimer's disease. And we can study this in mice because there are transgenic mice that have been manipulated that are kind of like Alzheimer's in human beings. So the question is, if we just had an anti-aging pill, would this, and if it prevents age-related diseases, might it be something that we could use to prevent Alzheimer's or other neurological diseases that occur with age? Now, as I often tell my group is, if you would have asked me about 3 or 4 years ago when we were going to get an anti-aging pill, and I've been in this business for about 40 years, I would always say, we're going to get there, it's going to take us about 10 years. I always use 10 years because it doesn't sound very far, and no one will remember 10 years, what I said 10 years ago. So I honestly felt that it would probably not be within my life, certainly not my professional lifespan. But I was surprised because about 2 to 3 years ago, we made a major discovery at the Health Science Center. And this was by 2 of our investigators. One was David Sharp. He came up with the idea. And Randy Strom was the person, the pharmacologist that essentially developed the formula for this compound. And what I'm going to do here is let this video clip tell you what happened.
At an exciting breakthrough in the fight against aging. San Antonio researchers used a drug called Rapamycin to extend the lives of mice, and someday it may help people live longer and better. Wendy Rigby has the story, new at 5.
At the Barshop Institute for Aging Studies at the UT Health Science Center, these tiny animals are stars today. They are elderly mice, more than 2 years old, equivalent to about 80 years for humans, and they're still going strong. They're part of an experiment using a drug called Rapamycin, encapsulated and blended into their diet to control a protein believed critical to aging.
The action of this protein actually becomes not so good. And so if we can inhibit it, we might be able to help slow down the aging process.
The special diet extended the life of middle-aged mice by 28 to 38%, a finding that was so remarkable it's published in the current issue of the journal Nature. That life extension would be greater than the predicted increase in human lifespan if cancer and heart disease were both cured and prevented.
There's a saying in geriatrics or in gerontology that we die of the first thing that kills us. So if we delay the diseases of aging, then that would extend lifespan.
Rapamycin is a compound made by bacteria in the soil. It's already used in people as an antifungal drug, anti-rejection medicine for organ transplant patients, and experimentally to fight certain cancers. But this is the first time rapamycin has been intentionally fed to mammals to see if it can create an anti-aging effect.
This target of rapamycin is, I believe, one of the principal keys of aging.
Another notable aspect of the experiment was the age at which the mice were treated, suggesting that even treatment late in life might be enough to prevent the onset of serious disease, increasing longevity.
The applicability to humans is still distant, but this was a significant step forward.
So, as Randy pointed out, it was a major, I would say it was a major step forward. This received a great deal of news coverage, and in 2009, the magazine Science, which is our top magazine in the area of research, named this as one of the top 10 discoveries for that year. Now, why was this so important? Well, it was 2 things. First is, this was the first time that anyone had ever shown that a pharmacological intervention, something relatively simple as a drug, but nothing like a genetic manipulation or a lifelong caloric restriction, where you could use a pharmacological approach to essentially slow down aging. It was the first demonstration, and the other thing that was so very, very surprising, which no one in aging, including myself, would expect, that it worked when it was introduced relatively late in the lifespan of the mice. In other words, the mice would have been like 50 to 60 years of age. I didn't mention it, but in the caloric restriction studies, we always started that when they were very young. And the assumption was, from several studies, was that if you waited too long, you would have very little effect. So what we showed here was a proof of principle that it was possible with a pharmacological, a drug-type agent, that we could have an impact on aging. And what I view this is much like in the 40s where they showed that antibiotics could treat infectious diseases. So it was a huge breakthrough. Now, of course, what we thought, well, since caloric restriction has an effect on most age-related diseases, we wanted to ask the question, was what about rapamycin? And we were fortunate about 2 years ago to get a major grant of about $5 million to study this in our mice. And the first thing that we wanted to look at was its effect on Alzheimer's. And the reason for this, as most of you know, is there is no treatment, no cure for Alzheimer's. And we thought, wow, this would be nice if this would work, and it potentially then could be taken to humans. So the two investigators, junior investigators that were involved in this was Veronica Galvan and Salvo, and they both did it independently in their own lab using slightly different mice. And we'll tell the story here.
It could be a major medical breakthrough discovered right here in San Antonio. The research that could change the lives of those with Alzheimer's.
Researchers in San Antonio have made a huge breakthrough in the search for a cure for Alzheimer's. A promising drug was successful in preventing Alzheimer's in lab mice. And as the Night Team's Jennifer Dodd reports, they hope it'll do the same thing in humans.
Mechanism of function. When Alzheimer's researcher Dr. Veronica Galvan heard a drug called Rapamycin being studied just down the hall from her was successful in lengthening the lifespan of mice, she wondered, could it also help patients with Alzheimer's?
That makes sense. I mean, if this is really delaying aging, you would expect that it would delay Alzheimer's.
So she decided to find out, launching a study of her own.
We're here at the labs at the Bar Shop Institute where these experiments are taking place. Now, they generally don't let the public in, but they let us come in to see how they did it.
Reinforcement, so it's a reward.
They used a control group where a number of mice were genetically engineered to develop Alzheimer's. When they reached adulthood, half were given rapamycin, half were not. They're subjected to memory tests using this pool of water. Now you can't see it, but there's a hidden platform under the water the mice have to find.
When they reach a certain age, which is 7 months of age, the ones that have been engineered to have Alzheimer's and could carry this gene, they will start having memory problems.
But what they found is that in the group given rapamycin, none of the mice developed Alzheimer's.
It was really remarkable.
Now, rapamycin is FDA approved to treat various conditions, but it has not been approved to treat Alzheimer's. Dr. Galvan says many more tests are needed before it's available to the public. At the Bar Shop Institute, Jennifer Dodd, KSAT 12 News.
Now, I just want to show you the remarkable data here. This is from the two studies. This is the one right here is from Veronica's study. This is from Salvo's study. And this is a measurement of memory. So the higher up these bars are, the better the memory. So you've got, this would be our control mice. These would be our Alzheimer's mice. And you can see that they perform about 1/4 as well. In other words, they remember about 1/4 as well. But interesting, when you give them rapamycin, they perform just as well as the mice that were not engineered to get Alzheimer's. In fact, Salvo has now done the study where, in this study, we looked at it about 4 to 5 months, which would be something like 10 years in human studies. But Salvo's taken his mice that developed this and has started feeding them early on and fed it all the way through, and the animals at the end performed just as well as so he was able to completely block the development of Alzheimer's. The other thing, if you remember, I talked about was the plaques and tangles that you see in the brain. Now, what you're seeing here, this orange colored stuff, is what we call A beta. It's the stuff that you see in the plaques. And you can see that the intensity, the amount of this orange-brown colored staining is dramatically decreased in the brains of our mice that were fed rapamycin. And this, the fluorescent, the green fluorescent measures the plaques, and you can see the amount of plaques. So not only do we improve or maintain memory in these mice, but as we would predict, it has an effect on the buildup of these plaques and tangles. I'm not showing you the tangles, which we think are important in the etiology of this disease. So the big question we have now is, will this work in humans? And so we're very interested in trying to essentially extend this to ask if we can use it in humans. Now, but you want to keep in mind, there's a caveat here, is the mice, well, you folks know this, is the mice are different than humans. And so the hope is that what we're seeing here in mice will translate to humans. But thus far, all of the studies where they've shown an effect of a certain drug or other things they've tested in mice haven't transferred to humans. Now, we think that rapamycin has a better chance than anything else for the following reasons. First, one of the things that's unique about rapamycin is that it doesn't just appear to affect Alzheimer's disease, but it looks like in other mouse models of other neurodegenerative diseases like Huntington's disease and Parkinson's, it looks like it might be beneficial too. So this might have a global effect.
Housekeeping, call 2342. Housekeeping, 2342.
So what we think the rapamycin is doing here, the way that it's working, is it acts as kind of a switch that turns on what we would call the body's garbage collector that kind of disposes of these protein aggregates, so that when you feed these mice rapamycin, it gets rid of these aggregates, and therefore you don't have the memory loss or the neurodegeneration. So it appears that it just doesn't work. It might not work just on Alzheimer's, but other diseases, which is unusual because most of these other drugs that have been targeted for Alzheimer's target just the plaques and tangles. Remember, we didn't target, we were just slowing down aging and seeing what effect it had. The other thing that's important to keep in mind is rapamycin is FDA approved for use in humans, and it's been used for quite some time, primarily in transplant patients with other immunosuppressive agents. And so one of our concerns when we were studying this is, would rapamycin be an immunosuppressant? In other words, reduce your immune system, because that would not be too great. Now, when people use rapamycin in transplant patients, they're using a lot of other chemicals that reduce the immune system. We've done, not me, but faculty at our institute have looked very much in detail at the immune function of our mice. And we can say pretty certainly that in mice, feeding rapamycin for long periods of time is not immunosuppressant. In fact, it upregulates some immune systems and downregulates others, but we don't think it has a major effect on the immune system. So we think that from the human standpoint, it's not going to be an immunosuppressant if it functions the same way as in humans. The other advantage here is that this is a compound that we think, will slow down aging in humans as well as animals. And why is this important? Well, this is important for two reasons. Number one, this means that it has an potential impact on other age-related diseases. Oftentimes you hear, remember, we get something for this condition or that condition, and then a few years later, after people have been taking it for several years, they said, oops, it causes heart attacks, or oops, it causes cancer. Theoretically, if we've got something here that's slowing down aging, it should have a positive effect on other age-related diseases. And we have evidence that that's the case. For example, cancer. We would predict that if you slow down aging, you'll reduce the risk of cancer. And certainly our studies, as well as other studies, have shown that rapamycin, when given to mice, will have an effect on cancer. This is just a study from one of our colleagues that's looked at pituitary tumors. And you can see that these animals will develop these types of tumors in the pituitary in about 12 months. And when we give rapamycin, we can see that it reduces the number of tumors dramatically, the size of the tumors, and the animals without rapamycin would all be dead within 12 months, and we can see that almost all of them are still alive. And we've seen it with other types of cancers. In fact, people are now using rapamycin with respect to humans in treating cancers. There was a study just this Christmas that made the news headlines where rapamycin was given to certain breast cancer patients to prevent the reoccurrence. And the study was so positive that it was terminated, and that was then used as the mode of care for the individuals. We're also trying to work with a group that's studying the use of rapamycin to treat patients that have had renal cell carcinoma. This is a cancer where it's primarily removed surgically, and there is a certain chance of reoccurrence, and they're treating patients with rapamycin. And we're very interested in looking at these patients with respect to memory loss and that sort of thing as well. So the other thing is, there's evidence that in mice at least, that there is a reduced risk of atherosclerosis. We have mice that are engineered so that they will develop these atherosclerotic plaques. And when we look at the mice that are fed rapamycin compared to the animals that are not fed rapamycin, you can see that there's about a 30% decrease in the atherosclerotic plaques. So not only would this have a beneficial effect on delaying Alzheimer's, but it certainly wouldn't induce your risk to atherosclerosis or cancer. In fact, it might reduce the risk. And the last thing that I want to point out is that if we have an effect on aging, we would predict that we should slow down the decline in physiological aging. In other words, the people should be more youthful. And we do have that evidence with respect to our mice. And here we've got, these are very old mice that have been either fed a normal diet, or is a mouse that same age, roughly like an 80-year-old person that was fed rapamycin. And you can see the animal on the right is much more younger looking and much more active. In fact, Steve has got his group where they're looking at various parameters of physiological function. And what they found is that while not all physiological functions are improved, the important thing is none of them are reduced. There's no evidence that the rapamycin has been detrimental, and many of the functions have been improved. So what would happen if rapamycin worked? Well, it would prevent or delay Alzheimer's. In other words, it would be a potential treatment for Alzheimer's. We would live longer, and we would be healthier at all ages. And so I want to emphasize this at the end, because I think this is a video clip I picked up, and we're not sure whether it's accurate or not, but it's been out 2 years, and no one has said, no, this is not a true video clip. Supposedly, this is a lady that's 92 years old, and she's dancing with her 29-year-old grandson. And what I'd like to say is that if we can slow down aging as we're predicting, this person, rather than being an outlier, would be the average. So take a look at this lady. I always say, I don't know when she's 92 years old, but if she's 72 years old, that's pretty darn good, you know. So as I said, you know, this certainly this person has, if true, has had the right genes, certainly has had the right lifestyle, and as I said, would be in our system would be an exception. But think of this, and it's not too far-sighted, is if we were able to manipulate the aging process, as we've shown that we can in mice, this would become the average. It wouldn't be an outlier. And so and that's what I want to emphasize. In other words, by altering the lifespan, we're not only increasing the amount of life, but we're increasing the life within those years. Thank you very, very much. And I'll be happy to answer questions. Before we start, I just want to mention is if any of you are interested in coming up and visiting the Bar Shop Institute, we'll be happy to have you. I'm speaking for Steve, but we'll be happy to show you around and have you meet with some of the faculty that are involved in this research and the other research. Any questions? Yes?
What is the percentage of mice experiments being proven to be helpful in adults? I mean, in humans?
Overall? Well, I think it depends on what you're looking at. In other words, certainly the one concern is the mouse models that have been used for Alzheimer's and ALS have, the therapies that have been used there haven't translated very well. But keep in mind, we don't have any other model. Now, my bias for thinking that why this hasn't worked very well is the mice that have been used here have been genetically engineered to overexpress beta amyloid that is found in these genetic families that cause Alzheimer's. And so when they're treating, in other words, the genetic forms of Alzheimer's make up less than 5% of the Alzheimer's that you see. So most of the Alzheimer's is what we call sporadic. We don't know why it occurs. But so these animals have been genetically manipulated to have. And so my feeling is that most of the people have been using beta amyloid blockers, and it works in the mice, and it may not work in the humans. The reason I think that our system has a leg up on everybody is we're affecting aging. And so since this would be a more diffuse process, I think it would have an effect on the sporadic. But the big problem is that there's the same problem with ALS in the mice. Many of the manipulations that work in those mice don't. And I think it's probably twofold. It could be that the animal is just more complex. And so one of the things that we're doing at the Alzheimer's, and Steve is heading this up now, is we're now testing rapamycin in a primate model system. In other words, we have one of the only aging primate center marmoset colonies in the world, and these are little primates about the size of a rat, but really cute little things. But they only live about 10 to 15 years, and so we can set up and do a study with these animals to look at the feeding rapamycin, and within 5 years we can get an effect on lifespan. Now, we don't have a problem with Alzheimer's in this primate model, but they do get amyloid deposition, so we can look at that. Steve?
Can I-
Sure.
If I could just add something to that. Another bit of evidence is that if we look at the treatments for cancer that have been developed in mice, about 1 out of 10 turns out to be effective in humans. So that's not great. But what we've done here is we're really not treating the symptoms, we're treating the underlying causes. And because we're treating the underlying causes, we're pretty confident that rapamycin is going to do it, but maybe it's not. Maybe it's a mouse thing. But the thing is, now that we're focusing in on the underlying causes of aging, we actually have 2 other drugs that are in the pipeline now. They're sort of partway through, and they both look as promising or even more promising than rapamycin. So even if this doesn't prove to be the silver bullet that it seems now, there's other things coming along, and so we're making really, really rapid progress in this area.
And I just want to reiterate, as Steve said, is the approach that we're taking to treating Alzheimer's is we're not going after amyloid. Because as I said before, there is a question whether amyloid is bad. You know, initially they thought the buildup of amyloid was bad. Now some people says, well, the buildup is good because the cells are storing it here, and the cells that died were the ones that couldn't do that. So we're not amyloid centric. In other words, we're from the age, so we're taking a different approach that we think has a better chance of affecting this sporadic disease. I certainly would love, I saw a TV special on a group down in South America that developed Alzheimer's. It's a genetic family, and almost everybody in the family would develop Alzheimer's by 50, and it would be really interesting to see what something like rapamycin would do. Yes?
How does the drug impact aging? I mean,
Oh, very good.
You're showing a cause effect, but
Yeah.
how does it?
Okay, so this is the other nice thing about rapamycin. I didn't go into the detail, but we pretty well know what rapamycin, or we think that we know what rapamycin is doing at the level of the cell. So it works on this a complex that's called TOR, target for rapamycin. But this is a pathway that actually was only discovered about 20 years ago. But this is how and it's found in all eukaryote cells, from yeast to human to mammalian cells. And what this is a nutrient sensing. In other words, how does your cell know when there's a lot of food around and it should grow, or the organism, and there isn't. And so this is a nutrient sense. So this TOR pathway, and I think I've got a little scheme of this here. I'll show you. So what you see here is in the cell is that if there are a lot of nutrients, primarily amino acids are the main signalers, or growth factors around, telling the cell and that the environment there's a lot of nutrients, and so it's possible you'll have growth and proliferation. And it's interesting because at the same time the cell downregulates this process called autophagy, and this is kind of where the cell essentially digests proteins and nutrients. So it's basically telling the cells, hey, there's a lot of food around, now's the time to grow and reproduce. And so what rapamycin does is it blocks this. And rapamycin is interesting because rapamycin is found in nature. It's produced by bacteria, and they think that the reason the bacteria produces rapamycin is because it tricks the fungi that it's competing with for food to thinking that there's no food around. So the rapamycin blocks this mTOR here, so that even though there's nutrients and growth factors around, the cell thinks that there isn't, and so it stops growing and it starts upregulating autophagy to kind of generate more nutrients for the cell. And so what we think that the reason rapamycin works with respect to aging is that with aging, one of the things that we see is that perhaps as we get older, growth and proliferation are not so good for us. And we can see cancer is the most obvious disease. In other words, and that's why rapamycin makes sense with respect to cancer, is that if you had cancer cells around and you had rapamycin, it would slow down the growth. We think the autophagy is important because we think that's probably at the basis of why these animals don't develop memory loss. In other words, and don't develop these aggregates, because we've upregulated this autophagy, which is kind of like the garbage disposal unit, and gets rid of those things. So we do two things. So at least those two things, and possibly it may have an impact on inflammation. That's kind of iffy. But that's why we think, at least I think, the rapamycin is having an effect on the age.
It's essentially a chemical caloric restriction.
That's what people were looking at, right. I think it's caloric restriction and rapamycin do some of the same things. I think caloric restriction does a little bit more, to be honest, but that's exactly why Dave Sharp came up with using rapamycin. He thought it was a mimic of caloric restriction. In other words, you could give this, the animal would think that it didn't have the nutrients, and basically that's what he had proposed. He proposed that almost 12 years ago, and everybody thought he was crazy. In other words, it sounded too simple. And it took about 6 years before we could get money together through this intervention testing program that we have at the Health Science Center to actually test this. And by golly, it worked. You know, this is one of those things that's very exciting that only occasionally happens. It worked, and as I said, not only did it work, but it worked late in life. Yes?
From what I'm hearing, like our children and grandchildren have to start using it at a younger age rather than us for it to do some good.
Not really. That's what's so exciting about it. If you would have asked me this 3 years ago, I'd say yes, but when would you have to start an anti-aging mechanism? I would say, well, you would want to do it probably in the 30s or something, pretty early, because my own, Steve may not agree with me, but my feeling is that aging starts roughly about when we reach sexual maturity, and then things go all to hell, But so my thought would be is that we'd have to do, if we were going to have an anti-aging thing, we'd have to do it early. This study with the mice would suggest that it would have an impact when we're talking about people at the age of 60 and 70. So, and that's really important because that always bothered me about the aging, because if you have to give a manipulation where you have to take it all throughout your life, you just have all the potential confounds. And this suggests that we would do it later in life when I think, certainly from the standpoint of growth and proliferation problems, we would see if you took rapamycin and gave it to an individual very early in life, that would most likely stunt their growth. But once you've maintained that growth, the size and everything, it seems to me that there's a lot of proliferation things that's happening with age that aren't necessarily good. And cancer, of course, is one of those, and this would kind of reduce it, yes.
Is there any pain associated with Alzheimer's?
Pain?
People have pain who have Alzheimer's.
No. Well, not physical pain as such, but I think it's, having gone through this with my mother, there's a great deal of emotional pain. And it's very sad. I remember 2 years ago when I went back to see my mother, and she knew that, it's interesting, my mother has never said she has Alzheimer's. She says she has trouble remembering. And it's not like she doesn't know what Alzheimer's was, because when her friends got Alzheimer's, she was always talking about it. But I remember going home about 2 years ago, and my mother said, I hope that the next time you come back to see me, I'll remember who you are. And so what I saw was she passed through a stage where, because she didn't know what was going on, she was just tremendously excited. I remember talking to her one evening, and this is when we had put her in the nursing home. And she said, I don't know where I'm at. I don't know who's going to take care of me tonight. And I talked to her and calmed down. Fortunately, as she's progressed, she's kind of lived, now has gotten used to not knowing. And as I said to my wife, who just visited her, my mother lives in one time zone. There's no future and very little past. And so I think for my mother right now, I don't see that there's any emotional or physical pain. She's very lucky because some people become violent, very difficult to take, so it depends on how they go through the stage. But from the standpoint of pain, there isn't. But from the standpoint of emotional pain and seeing a person lose their faculties that make them human, their mind, is very difficult. And as I said, it's, if I believe, my mother's 96, I believe that if she hadn't had Alzheimer's, she would still be able to be at home. She's frail, and we'd have had to have people come in and take care of her, but not have to put her in a nursing home. Yes?
Is there any relationship as far as heredity is concerned with Alzheimer's? If your grandfather had Alzheimer's, your father had Alzheimer's?
There is a heredity component to it. Yes, there is. But the major component is the age. So do you, on the heredity, do you have anything?
Yeah, well, there are 3 genes that if you have them, you're going to get Alzheimer's disease, probably in your 50s. That's very rare. There's another gene, one of the ones that was up there, called ApoE. If you have that, you're at increased risk. So if 3 or 4 people here and there in your family have had it, but not everybody, it's quite likely you might have something. And then there are no doubt other genes that have smaller effects that we simply don't know about yet. Okay, so most of the cases we know about, it's not hereditary.
But the ApoE gene, the variant, it's interesting because I was talking to my physician. I go in and see him, and we talk more about my research than my physical condition. But he was, I was telling him about the rapamycin, and he says, "Wow, I just had a patient that came in that had this ApoE gene," and he said, he told her, this is an 80% risk, so it's not nothing, and so there are and he had said, well, I'll mention to her about the rapamycin, but the one difficulty in, setting up a trial to look at, let's say, the effect of rapamycin on, let's say, Alzheimer's, is the difficulty is you need to get people before they're full-blown Alzheimer's, because the general feeling, and we've got some studies with mice that suggest that if you wait until the plaques and tangles are already developed, it's going to be impossible to reverse it. You kind of slow it down. And so it's believed that Alzheimer's is one of these things that when you see it, it's probably been progressing for 10 or 20 years before that. So it would be important to essentially identify people when they're show those first signs of cognitive impairment for a treatment.
Don't you already have a group of people that you could study that are taking this drug?
Yeah, exactly.
Are you looking at this group and comparing it? I realize that's not a perfect control group. But
You hit it right on the nail, right on the head. In fact, we've talked about trying to do rapamycin studies. It would be very difficult to do a clinical trial where I would take you folks that are fine and take rapamycin, because there are some side effects to taking rapamycin, so it'd be very difficult to get that through. But as you pointed out, there is a group of people that are taking this, and these are cancer patients. And so the kind of strength, and this is where science doesn't always talk to each other, is there is this study, the Everest study, the one that I was telling you about, where they have renal cell carcinoma patients, where they've removed it surgically. They haven't been treated with chemotherapy or anything, and they're giving them a rapamycin type compound. And what they're interested in is a reoccurrence of cancer. And what we're saying is, hey, we would like to piggyback on this study and look at what's happening with respect to memory, cognition, immune function, health status, and a variety of things. And so we're right in, we've been talking about, Steve actually was involved in initiating this study, and we're working with our clinical colleagues as well as Ian Thompson, who came down here a couple months ago to do this. So that is the perfect way, I think, to set up and do a study like this. And I think what's going to happen with the breast cancer study and everything, I think rapamycin is going to become more and more interest to the cancer peoples, as not so much as a way to essentially, once you have the cancer, to get rid of the cancer, but essentially prevent that cancer from coming. So that we've talked about this before, is if you're at a high risk for breast cancer or colon cancer, taking rapamycin might be a way of preventing those little cancer cells from growing and dividing. You had a question?
What are the side effects of rapamycin?
So the side effects, about 80% of the people will have sores in their mouth. There is I forget what it's called, but it's like a pneumonia in the lung. It's not inflammation. But both of these can be, in other words, if you develop these problems, the rash and the mouth problems are more just, are not hugely a negative problem, except it's something difficult to live with. But if they take people off of rapamycin, these conditions will go back so that they're not there anymore. And then they put them back on rapamycin, and usually by going through this a couple times, then you can get over that hump.
So the sores in the mouth, and what else?
It's the sores in the mouth and a type of pneumonia. And the pneumonia one is the big one. But the cancer people can follow up, and you can detect it once you, as I said, as you take them off of the rapamycin, these things will come back. Yes.
There's one other you're forgetting about, and it's important because we started this in basically 60-year-old mice and had this effect. One of the effects that it has is it suppresses testosterone. So your 30-year-old is unlikely to be interested. Your 30-year-old male is unlikely to be interested. Your 60-year-old male, on the other hand, who cares?
Wait a minute.
the other thing, the other thing that we found is that there may be, and this wasn't reported, well, it's kind of been reported, but it's not, but in our studies, is there may be a tendency for these animals to become insulin resistant, and that would potentially lead to diabetes. And so that's one of the things that we want to study in this. And so as I said, is, you know, while there is some upsides to rapamycin, there are downsides. But if you're looking at this thing, having lived with my mother, if I'm looking at these things, and this is what you're going to find almost with any compound. In other words, it's never going to be all good. But as I said, is the beauty of the rapamycin is there is a history of having people on rapamycin for like 6 to 10 years. And so we have a lot of information. There was another question, right?
Yeah. Is there any indication that any particular profession is more dominant as far as this is
Likely to have?
Yeah.
Politicians.
Well, what I've been concerned about was like contact sports or...
Oh, okay. No. So the one, and I noticed you didn't have the risk factor of this. So one of the certainly risk factors is head trauma. Head trauma. So you've heard a lot with respect to football. But the one thing, as was pointed out, I'm associated with the VA. And one of the things that we don't talk about is all of our veterans from the Middle East coming back with head trauma, which is the major wound in this war that we've been in. And so we're very concerned about, you know, getting them through this. But what they don't realize is these individuals are going to have an increased risk of Alzheimer's. And so one of my colleagues, Holly, is very interested in looking at using rapamycin and seeing if it could be protective with respect to brain trauma in mice. So that's kind of one, it gives you a feeling for how the research in one area where we really started to ask the question about, did this have any effect on aging? We've suddenly, now that we've done this, it's come out and we're very interested in how it impacts various types of diseases that could have an important, so that in this case, so the problem, as I pointed out with Alzheimer's in using rapamycin, is we don't know who's going to develop Alzheimer's or anything like this until it kind of, I'm on the board of counselors at NI, and so there's a big program to try to find those things that will tell us the increased risk factors for Alzheimer's. We don't really have any good, but certainly in brain trauma would be a totally different thing. So if you had brain trauma, you would know that you might want to take rapamycin to prevent some of these things, right? Any other questions? Yes.
So, I mean, I think you just answered part of this, but red flags, I mean, when do you just say, laugh off that you're being forgetful, and then all of a sudden,
that is a very good question,
yeah,
because I thought, during this talk, I thought, you know, I used to be much quicker. I could come up with these terms quicker. It's very difficult. Now, the physicians that look at Alzheimer's have tests that they can use. But the big question that I had is, you know, now they're talking about mild cognitive impairment, you know, and there's actually a group in Seattle, Washington, who's has a study they're going to try to start using rapamycin in people that are mildly cognitively impaired. And so we'll see, but it is really very difficult to diagnose. And I know that totally from my mother's case. You know, it's like a year, we thought everything was all right, and then suddenly we noticed. And there must have been things that were going along, but we didn't just pick up on it. Do you have anything on the diagnosis?
Well, the Alzheimer's Association webpage has a list of symptoms that you should be alert for if you're concerned. So if you went to the Alzheimer's Association, it's got a very nice webpage. Just sometimes you wonder. I mean, everybody forgets stuff, right? And you think, but there are other things that they can use. So I would suggest you do that.
Yeah, but it is tricky because, you know, when this happened to my mother, I'm just constantly asking myself, you know, when you forget something, and you start getting up in years, you say, is that happening, you know? And some of this is normal, and it's not Alzheimer's. Yes?
Like, for instance, nowadays, in recent years, when you have medications that you take, you get a copy of all the side effects of things. Right? If a medication says it may cause memory loss, is that memory loss they're talking about? Is that the same as leading to Alzheimer's? Or is that totally
probably different. Probably different. And you kind of, my wife's a great one for reading it's things on the medicine, and my feeling is now it's got almost any possibility, and so they're just kind of, and so it's really very difficult unless you go in and look at the literature to know how often does memory loss or something happen. But the memory loss that you would have on a drug would probably not be leading or causing this. That's would be, in most cases, that would be, because my guess is that if they had shown this, it would be very difficult to market that compound.
The drugs now that are being prescribed just to sort of help limit symptoms, is it Aricept, and what are those drugs doing for?
I can't remember, I don't know if you know, Steve, but my feeling is that many of them are trying to upregulate some of the neurons that remain. And so I know they used to have the cholinesterases are one part of the brain, and so they tried to kind of upregulate the kind of neurological activity in the brain. But talking to my clinician, geriatrician friends, is that most of the time these are not particularly effective, and oftentimes they have side effects that are even worse. I have a colleague who had his wife tried some Aricept, you know, and it had more negative effects than it had positive effects.
And is the rapamycin generic?
Okay, good question. So the rapamycin that we're talking about this is the compound that was initially discovered and marketed. I believe the patent is off on that. So what you have is you have a variety of Rapalogs, we call them. And I think I've got here a slide that would, show them. So there are a variety of these that have been chemically synthesized to make the drug a little bit more available. It's my feeling on these compounds is that these are all working the same way as rapamycin, but they have two things: is they make it a little bit more available than rapamycin. Because one of the things that I didn't tell you about was Randy Strong did a huge effort in this whole thing, because we were feeding rapamycin to the mice. And so if you're going to do this, most people before that, when they gave rapamycin to mice, they would inject it into mice. Now that's fine if you're doing a study of about 2 weeks. But if you're doing a study over 2 years, the mice don't like it, and the people that you got injecting it don't like this. So the obvious way to do any studies on aging with the drugs that we've been looking at is either put it in the water or in the food. And so this is a compound that's not water soluble. And so we put it in the food. And the problem is that it's relatively unstable. And so when we put it in the food, he couldn't get the Rapalogs levels in the blood up unless he used large amounts of it, and it was so expensive it didn't look like they were going to be able to afford the study. So Randy went and looked around, and he came up with the idea of microencapsulating the rapamycin. Then it would be stable there. We put it in the food, and the capsule would dissolve in the intestine, and it would be taken up by the animal. So that was a novel discovery that we're trying to patent right now. So it's a different delivery of the rapamycin. And so, and this is why we did the study, and we started it late in life. We'd never done the study, but we had all the animals, and we were going to put them on the study, but we didn't have the rapamycin in the chow. And so by the time that they developed this encapsulation method, the animals were 19, 20 months of age. And they were sitting there what to do. And as Steve and I often say is, we'd probably sit there, well, forget about it, get rid of those mice, And they said, well, we'll give it a shot. And I remember when Randy told me, he says, well, I said, well, good, but get the other study started right up, because this one's not gonna work, and we did you know, it was serendipity. So that what you've got here, so we took the rapamycin, or the sirolimus, this is the one that was developed originally. This is the kind of the pharmacological name. This is the one that's not very soluble. So when they give it to people, it's kind of a slush type thing that they drink. Now, since they have developed a variety of other types. Everolimus is the one that's being used in the renal cell carcinoma. This is one that's relatively new that was developed. And so it's my understanding that all of these have about the same impact on the mTOR system that rapamycin does. But these things are different ways of delivering it, number one. And then the other thing is these things are now under patent, whereas the other one's not. So one of the interesting things is that rapamycin theoretically is a relatively cheap, generic way of getting the same thing. So if you're out looking to be treated with rapamycin, you're going to be using these types of compounds. Any other questions? Yes?
As you speak about this for Alzheimer's, it also has strong implications for all the dementias and
Yes,
those two,
yes,
right?
yes. We're very interested in, one of our faculty members looking at this in Parkinson's. And right now, the data aren't as striking, but there are some studies out there. But again, the Parkinson's mouse model is even worse than the Alzheimer's mouse. But there is some very high-profile study on Huntington's disease, where it had an impact. So I'm very optimistic that this might have a generic effect on those types of neurodegenerative diseases that have this buildup of protein glops in the brain.
Like
That this
Lewy body.
exactly.
and
Yes. Yes, and that's the other thing that I think we have going with rapamycin over these compounds that are targeting the beta amyloid, is that we could be, you know, if beta amyloid has nothing to do with it, I mean, we're upregulating and getting rid of theoretically other things too. Yeah. Yes?
That has nothing to do with your study, but this big boom about coconut oil being the magic cure for Alzheimer's. What about that?
I don't know much about it, but it doesn't make any sense to me. Is it to you, Steve? No, it's not. It's not something, I have heard this more on YouTube than through the scientific, the medical journals. So it, you know, my sister actually sent this to me and says, "What do you think of this?" And I wrote back to her and I says, "BS." Yeah, but yeah, it's one of those things. It's too good to be true. You know. Any other questions? Well, thank you very much. You've been a great audience.